This is screen capture video 12.4, and in the last video I was talking about the messenger RNA molecule moving out to the ribosome and attaching to the ribosome and moving across the ribosome. And as the messenger RNA molecule moves across the ribosome, each three bases, which serve as a codon, are primed so that they are read. Keep in mind, the ribosome is made up of RNA and protein. There's a large subunit and small subunit. Excuse me. The transfer RNA molecule is going to be coming in, and attached to it, it will have an amino acid. And each transfer RNA molecule brings in a specific amino acid. The transfer RNA molecule, somewhere along its length, will have an anticodon of three bases. That anticodon is complementary to the codon on the messenger RNA. When these two transfer RNAs are attached to the messenger RNA as it's moving across the ribosome, the amino acids are close together. They can be bonded with a peptide bond, and the amino acid comes off of the one transfer RNA, and the next transfer RNA can come in, and the messenger RNA molecule moves along across the ribosome, and the next three bases will be read. The codon, the transfer RNA will come in with its anticodon, attach, and two amino acids will be close together, and so you get this lengthening of the amino acid chain. It's called polypeptide, and it just grows as these transfer RNA molecules come in with their amino acids. Over here on the right, it shows it in a more simplistic way, but you get the idea. The messenger RNA is moving across the ribosome. The ribosome is shown in the brown here. Two of the codons in the messenger RNA are primed so that they're ready to be read and attract anticodons. Transfer RNAs come in, they attach, and the newest RNA coming in with its single amino acid has its amino acid close to the previous chain, so those are bonded together. When those are bonded together, it shows us a little bit further here. The transfer RNA can leave. The chain is still growing. The messenger RNA moves along the ribosome, and the next three nitrogenous bases, which serve as a codon, will be primed and attract another transfer RNA molecule. This occurs all along the length of the messenger RNA molecule until it comes to a stop. It says stop the process. And all the while, this polypeptide chain is growing, getting longer and longer. As it's growing, it automatically folds into a secondary structure, and then eventually a tertiary structure, and eventually two or more tertiary structures will attach to each other and form a quaternary structure. Okay, so here's the transcription process shown in this illustration. There's the translation and the polypeptide chain that's formed from translation and eventually protein synthesis. The folding is part of the protein synthesis. There's a video link here for translation, and you're welcome to look at that if you want, or find another one that explains it properly. So we have information that moves from DNA to messenger RNA to the protein, and the protein is what we see as the physical expression or the phenotype of an individual. Sometimes in the past I've used a computer analogy with the hard drive symbolizing the DNA, the chromosome. It's huge, it's bulky. If I were to ask a student to bring in a report and they brought in their hard drive, their tower, I would say, no, that's not what I need, that's not what I want. I don't want to carry a bunch of towers around trying to extract information. There would be sometimes when bringing a jump drive in would help. That you could say that might be symbolic of a gene, but still what I would prefer is the printed document or a file that would be sent to me, and that would actually be the physical expression or symbolic of the phenotype, the protein. Okay, some of this is just a little bit of review with the illustrations I have. Sometimes I'll use these as I talk about a little bit more about the flow of information. This does bring us to a discussion about mutations, because sometimes there are problems in the reading of the DNA of the gene and how it is transcribed or translated. And it usually begins back with the DNA. And a mutation you can think of as any change in the sequence, the base sequence of the DNA, because again, it is the bases in the DNA that serve as the code. It's not the phosphate sugar backbone. That's repeating. That's not significant as far as the message. That just is holding the bases in place and allows the message then to be transferred. Here's what's been, and up above here, anything that causes a mutation is called a mutation. And examples would be ultraviolet light. If a person were to expose their skin to ultraviolet light by tanning parlors, tanning beds, or by sunlight, repeated exposure to sun. Farmers, people are outside a lot that are older, have problems with this, sometimes not so much older. I've known of people in their 40s and 50s to have melanoma and die from it. Unfortunately, I know one person that was laid out in the sun quite a bit, and I believe in their late 40s, maybe about 50, died of skin cancer, unfortunately. X-rays. It's thought that the X-ray crystallography that Rosie Franklin was performing may have contributed to her ovarian cancer. She happened to be Jewish, and she was from the Ashkenazi line of Jews, if I remember correctly. And I remember reading on the internet that the Ashkenazi Jews tended to have a higher incidence of gynecological cancers, interestingly. It seems to follow in their genetic line. Madame Curie is another one that comes to mind. She worked with radioactive substances and won Nobel Prize with her husband, Pierre Curie. And if I remember correctly, she miscarried at least once, maybe several times, and eventually died. And it's thought that her cause of death may have been attributed to the radiation. That would be beyond X-rays. That would actually be radioactivity, the gamma rays, which are another mutagen. Chemicals can serve as mutagens. Formaldehyde has been implicated. Carbon tetrachloride used to be used in chemistry labs. Generally, it's not used anymore because it is considered a carcinogen. Asbestos, some of the chemicals in cigarette smoke, even alcohol, has been described as a mutagen. So there are a number of different things that can serve as mutagens and change the structure of DNA. Here they're showing the normal codon, it is the normal codon that forms from the normal hemoglobin. And there's a glucine that forms at this particular location. In some people, and it seems to be in groups from African origin and from the Mediterranean area, southern and eastern Mediterranean area, where in past times, historically, and even today, there have been problems with malaria. It seems that the malaria helped select for people who had a mutation in the hemoglobin and had sickle cell anemia. And so people of African origin tend to carry the gene for sickle cell anemia at a higher rate than other groups, and then some of the Middle Easterners, people in the Mediterranean area. And that might seem strange, but when you think of Syria and Iraq, they're desert-like countries, but they have some big rivers running through them, especially Iraq with the Euphrates and Tigris River that runs through it. Also, if you remember your history, when Rome was settled, they drained the swamps in the area of Rome to decrease the mosquito population, and that seemed to also decrease malaria. But what happens is that there's a mutation where there should have been a thymine, there's an adenine in the DNA. That codes for a uracil instead of an adenine in the messenger RNA. So you have the wrong codon, which codes for the wrong amino acid. That single amino acid in hundreds will cause the hemoglobin to not form correctly, and it changes the shape of red blood cells, and they're more sickle-shaped, and they don't move through the blood vessels as well, especially the capillaries, because the red blood cells are being squeezed through the capillaries under pressure. And so if you have these sickle cells, they tend to clog up and causes extra problems for a person, and causes extra pain, and usually people with sickle cell anemia don't live as long. And that's just one location. That's actually a point mutation or substitution, base substitution. Here again, showing the base sequence for a normal gene. I wish they showed T in there. Excuse me, this is for RNA, so it's not T, it's the U. I wish that, what they did up here, that's good. You see the A, U, G, and there's a C, so you see all 4 letters in the messenger RNA, and this codes for this sequence of amino acids. This is considered normal for this hypothetical situation. If you change one of these bases, and this would actually be changed back in the DNA, but for simplicity they're just showing it in the RNA, change this G to an A, that is called a base substitution or a point mutation. I'm not sure which way I'll put it on the test, but that's in one single location. Generally what happens, that only changes one amino acid. Now how drastic is that? Well, it can be drastic in the form of sickle cell anemia, or it can be unnoticeable. It depends on how it affects the folding and the shape of the polypeptide chain. It might not even change the amino acid because of redundancy in the code. The new codon that's formed may code for the same amino acid. But if it does code for a different amino acid, it depends on how it changes the shape. If it doesn't change shape, not much effect at all. And that's something that geneticists have found with the Human Genome Project in the last 20 years, that different people have different sequence of DNA for the same gene. And it just so happens that the mutations that have occurred haven't affected drastically the expression of the gene. A type of mutation that's more severe would be a base deletion or a frame shift mutation. Again, know each of those terms. It's base deletion according to your text. Another text by the same textbook company that produces the biology text would call it a frame shift mutation, and I think you'll understand when I explain this. If you delete a base or add a base, it could be a base addition, which will cause a frame shift mutation. That shifts all the bases over one, because it takes 3 bases to make up codon. And if you remove one of those bases, then it's the next base that moves up to form the codon. And that generally causes all of the, or most of the amino acids to be coded incorrectly from that point on, downstream, so to speak. It's called frame shift because you're shifting the whole frame of bases over one, because you need 3 bases for every codon. That's a more drastic mutation. Just to mention one mutation that's somewhat interesting and tragic in humans. In animals, not so good for them, and that's albinism. Albinism in humans will cause pale features, very light hair, light-colored eyes. When I was at the university, I was told by an instructor that you could tell a true albino because they had pink eyes, whether it's animal or human, but animals generally is what we were thinking of. I've come to find out maybe the information has changed and they've modified the definition of albinos, but they have light eyes, could be gray or very light blue. Some of those groups of people will be included. I'm about out of time, so I'll continue this discussion in the next video.